Hey guys, welcome back to my channel Anytime Medicine, and today we are going to talk about is pyrimidine metabolism. So firstly we are going to talk about is pyrimidine itself. They have role in DNA and RNA. There are three pyrimidine structures: cytosine, thymine and uracil. Thymine we can see in DNA, uracil we can see in RNA. Methylation of uracil makes thymine. So what is methylation? Adding one CH3 group in any structure, that is called methylation. Unless purine, pyrimidines are one ring structure. Down below you can see cytosine, uracil and thymine structure. On your right you can see the basic pyrimidine ring, and on your right box there is carbon and nitrogen which are come from aspartate. On the left box you can see the carbons that comes from carbamyl phosphate. Now we know the purine and pyrimidine, so now we will learn the bonding of this nucleotide in the DNA structure. In DNA, specific purine bind with the specific pyrimidines. First bond cytosine with guanine, which is called CG bond. The second bond, which is adenine with thymine, which is called AT bond. Now CG bond has 3 hydrogen bond and AT bond has 2 hydrogen bond. So CG bond will have increased melting temperature for DNA. Certain bacteria have series of CG bonds, so that will have high tolerance for temperature. Now we are going to talk about pyrimidine synthesis. In order to synthesize pyrimidine, we will need ribose 5-phosphate, amino acid and carbons like we have already seen in purine synthesis. We will need the same structure as purine in the pyrimidine synthesis. Our goal is to create CMP, UMP and TMP, which stands for cytidine monophosphate, uracil monophosphate and thymidine monophosphate respectively. The first pyrimidine synthesis we are going to see is uracil synthesis by dividing into 3 steps. The first step is to make carbamyl phosphate. We can make carbamyl phosphate from glutamine with the help of ATP and carbon dioxide. We will need also enzyme carbamyl synthetase 2, and if we have too much UMP, that will give negative feedback to this enzyme and that will slow down the reaction. Our second step is to make orotic acid. We can make orotic acid from carbamyl phosphate by just adding the aspartate group in it. The third step is to make UMP. Orotic acid and PRPP will react with each other and make uridine phosphate with the enzyme UMP synthase. The PRPP stands for phosphoribosyl pyrophosphate, and here you can see the structure of uridine monophosphate. So the second pyrimidine group is cytidine. For cytidine synthesis we will need uridine monophosphate that will go under phosphorylation and will create uridine triphosphate. In order to create cytidine monophosphate, we will have to remove one oxygen group from uridine and we'll have to add one nitrogen group into the process. So we will see the structure of cytidine monophosphate that will have one nitrogen group instead of one oxygen group. So the third pyrimidine is thymidine. Thymidine synthesis we will going to divide into 3 steps. The first step is to convert UMP to DUDP. Uridine monophosphate will go under phosphorylation and will create uridine diphosphate, and uridine diphosphate will create deoxyuridine with the enzyme ribonucleotide reductase. So the second step of the thymidine synthesis is to convert DUDP into the DUMP by removing one phosphate group, that is called dephosphorylation, and the third step is to convert DUMP into the DTMP. We will do that by enzyme thymidylate synthase and N5N10 tetrahydrofolate. N5N10 tetrahydrofolate is the one carbon source for DTMP. So the DTMP will have one extra added carbon into the structure from DUMP. So the question is, from where N5N10 tetrahydrofolate comes from? It comes from folate. Folate we consume from diet, convert into dihydrofolate, and that will convert into tetrahydrofolate. The tetrahydrofolate convert into N5N10 tetrahydrofolate by doing some series of reaction. Here you can see the structure of N5N10 tetrahydrofolate, which itself contribute for thymidine synthesis. Sometimes folate directly will convert into tetrahydrofolate by enzyme of dihydrofolate reductase. We will going to see some abnormalities and drug related to dihydrofolate reductase in my next video. On your right, you can see the structure of folic acid, dihydrofolate and tetrahydrofolate. Thank you everyone for watching this video. Do subscribe my channel and share it to your friends. See you until my next.